that, and also today we will go a little bit deeper into the look at the history and then move forward with the the present actually. So we'll talk about how it was done earlier and now what are the changes and how AI is doing all those speedy things actually, how it is helping us to discover in new drug and all those things. So if you look at the pre-industrial drug discovery, like if you go back like 5,000 years or 10,000 years back, so historians and especially archaeologists, they have found that the seeds of betel nuts were found at spirit caves of northwest Thailand, and then this betel nut is supari actually, which we call in Hindi supari. So this betel nut was found in the caves. So it indicates that people were using that betel nut, and because betel nut is a kind of a mild psychoactive agent. Psychoactive means it activates your brain. So it activates your activity level, activates your brain. So it's kind of psychoactive drug. And then during 7,000 to 5,000 BC before Christ, the use of hallucinogenic mushroom is also implicated by Saharan cave drawings. So they were in Sahara, there were caves, and in those cave drawings, it was shown that people are using hallucinogenic mushrooms, and we use mushroom for making curries and all, but those mushrooms are eatable, but there are mushrooms which are not eatable, which are toxic and which are, they can be poisonous, they can be toxic, and some of them, they have these medicinal properties as well. And in 2680 BC, lime-containing betel nut shell found in Duyong Cave of Philippines. So means that is, the lime, the tuna, with the betel nut. So the lime extracts the compounds which are present in the betel nut. So they help them extracting them out of the fruit, the nut actually. So it was found in Philippines, and then he knows that alcohol has been used for recreational or medicinal purpose for centuries. So the early documented evidences, these are a treatise of medical diagnosis and prognosis, which is a documented medical method and prescriptions on stone tablets by Mesopotamians in 1700 BC. And then there was this Ebers papyrus by the ancient Egyptian. It contains several hundred prescriptions for the treatment of diseases or symptomatic relief in 1550 BC. And we presume that Ayurveda is also at least like 2,500 years old actually, coming from the Vedas. So the basic principle of prehistoric drug discovery was that because we were living with the plants and the animals in the surrounding and the environment, so we were depending almost exclusively on plants, plant-derived mixtures or plant extract. And that is how, those traditional methods like Ayurveda, Unani, Siddha, they could develop. And the medications were identified using empirical observation of the presence or absence of symptoms in patients rather than an understanding of the disease or condition afflicting the patient. So as we say in Ayurveda that we are taking a holistic approach towards the treatment. Means we are not bothered about the symptoms actually, but we are treating the disease. Okay, in Ayurveda. So that is called as a holistic treatment, where, for example, if there is a patient is suffering from infection, so he will get fever, and then in Ayurveda we don't bother whether it is a kind of which kind of bacteria or virus or any other microbe is causing that disease. So we are interested to know how the patient can become, better, so they can feel good. And this was observed by trial and error method. So people, when someone got sick in the old time, so they used to eat, okay, this plant looks like bitter. You can, if I try to eat it, and we'll see what will happen. And this also led to one of the Nobel Prize in medicine for the discovery of artemisinin. So artemisinin is an anti-malarial drug, which was discovered from the Chinese Materia Medica, Chinese traditional therapy books. And then the discoverer was Tu Yu. So, she did experiments on herself. So she got infected with malaria, and then she was eating that plant extract, using that plant extract to see whether she can get recovered or not. And that led to development of the artemisinin. So most of the efforts were performed in the absence of the vast majority of the fundamental knowledge required to understand even the basic principles of disease progression. So yesterday, if you remember, we talked about those pharmacokinetic properties, like how a drug is going inside body, how it is binding to the receptor, to the target. But in earlier days, it was not like that. It was just, we were interested in knowing the result, the outcome. So whether something can treat some disease or not, it was not like how it is binding, where it is going, how much of that is there in the body, all those things, because we did not have that kind of basic understanding, molecular biology level understanding of the disease and the targets and the drug discovery. So there is a very, very interesting story of discovery of quinine. So quinine, maybe who will tell me what is quinine? What is it used for? No, quinine, malaria?
No, sir. No, sir.
Malaria, treatment of malaria, right? Quinine. So people, so in dispensaries, you will get quinine tablets, which you have to eat if you get infected with malaria. So malaria is, a parasitic disease, which is caused by mosquito bite. Means that parasite is lives in the body of mosquito, and then when it bites, so it get transferred from the mosquito to the person actually. So Agostino Salambrino, who was from 1561 to 1642, he observed that Quechua is a kind of a tribe. So Quechua is a tribe. So Quechua people, whenever they were getting sick, so what they were doing is they were chewing the bark from the cinchona tree in an effort to relieve shivering and fever. So whenever they are getting fever and shivering, so what they were doing is they were just like taking the bark of cinchona tree and then eating it. So the febrile phase of malaria might be positively impacted by cinchona bark. And what he did was, so he took a sample of this cinchona bark, and then he sent to Rome for evaluation of it as a treatment to malaria. And then finally we got the quinine. So this is on the right hand side, you have the structure of quinine. So we got the quinine identified, discovered, and therefore Quechua people became the source of first successful anti-malarial agent, a drug that was a first line treatment for malaria infection until 2006. And later this quinine got resistance against malaria. So now this become this is not a good drug to treat malaria. So then you need new drugs actually. So then you have artemisinin and other drugs, other therapies. Likewise, this is a foxglove plant. Foxglove, and then this plant is a source of digoxin. So this is a glycoside, a compound which is used to which is used to treat the cardiac arrest. So if someone is suffering from some heart attack or cardiac problem, so doctors, they prescribe this drug to them, actually, it is digoxin. So this is a drug to treat the cardiac arrest. And then there are other examples like morphine, cocaine, aspirin. So who will tell me where does the morphine come from? Morphine? Where does the morphine come from? No?
One question. Hello?
Sorry? Yeah.
Sir, one question is that in case of medicine, it has some side effects. The allopathy has side effects. But whether plants are also, Ayurveda has also some side effects. Because if cardiac arrest can be resolved through that plant, then whether we can take that plant daily or it has some side effects for that.
Yeah. So, so that is a big myth, actually. It is a big myth that plants or the Ayurvedic drugs or if something is natural coming from plant is lack of side effect. Okay, because it doesn't matter whether the compound, the molecule, for example, aspirin. So this is a compound, right? A molecule. So it doesn't matter whether this aspirin is coming from plant origin or the synthetic origin, whether we make it in the laboratory or a plant is making it in their, how to say, their bark or their leaf or their body, I would say. So it doesn't matter. The structure is same, the same way it will act on the body and then it will have the effect. The difference is, for example, when we take a single drug that is in pure form, okay, in pure form, and then that single drug you need in small quantity to have the effect in the body, okay, and then sometimes if you take it in a large dose, it will have some kind of side effects. On the other hand side, if you take a plant, for example, if you just take some leaves and then or bark or something, so those barks and leaves, they have a mixture of compounds, so they do not contain single compound. They have a mixture of compounds relatively at low concentration. Okay, so when you will need large quantity. One thing is that you will need large quantity. Another thing is that when it goes into your body, so you don't know which compound is working actually. So it might be that all the compounds which are there in this plant, they have similar kind of effect, and they're showing that effect and working on the body. So it is like that. So it is a myth that the natural, if it is a natural, then it is non-toxic or it is non, So for example, cyanide is also a natural compound, right? Cyanide is also a natural compound. And snake venom is also a natural compound. So, but they are toxic. So botulinum toxin, which is the most deadly toxin, which can kill, a very small amount can kill a big elephant as well. So it is a myth that these compounds which are coming from plants, they are not toxic. The only thing is that they are not pure, so they do not have that much quantity to elicit the effects. I hope this will help you.
Hello, sir. I have also one question related to this discussion. That is the homeopathic medicine have the side effect, because they are also prepared from the different like a plant extract or animal extract or the different types of extract. but allopathic medicine, homeopathic medicine have very little side effect compared to this. So can you bring light on?
Again, I will say the same thing. Because yesterday I told you that everything is toxic. Everything is toxic, including water. Water, we are 70% of our body is made up of water, right? So if we drink 20 liters of water in 1 hour, we will definitely, the person will die because of excessive hydration. So everything is toxic. The toxicity depends on the dose. So in allopathic and as I said in homeopathic as well, when you are using an extract, so that extract may contain 1,000 compounds. For example, if you make an extract, those who are from chemistry background, so they can understand it properly. So if you make an extract of tulsi leaves, so you will find at least 100 different compounds in that extract. Okay. And when you are taking, so you do not know which of those compound is acting, which of those compound is giving you the required effect. But on the other hand side, if you take allopathic, because those are in pure form, so they have high potency, means they can cure the disease quickly, but then of course they will have side effect as well. So everything which goes into our body and does not belong to our body will have some or other kind of effect, because they cannot be highly selective. They cannot be they cannot go into our body and just bind to one cell and not to the other, and that is how this side effect comes from. So usually those homeopathic medicines, they are they works on the basis of natural immunity, like it says like poison kills poison, something like that. On that principle they work, at least the homeopathy. And Ayurveda has another approach, so where they have these 5 components, Vata, Pitta, Kapha, all those things, and then they correlate with that, whether this disease is caused by increase in the Vata or Pitta or Kapha, and then they focus on that and treat that rather than the disease. So these are alternative therapies which are extensively used, especially in India and other Asian countries. Okay, so coming back, yeah. So when we come to modern drug discovery, so Paul Ehrlich was the father, considered as the father of modern drug discovery. So he got a Nobel Prize in 1908 for his discovery of magic bullet theory. So he said that differential affinities of biological tissues for different dyes such as tryptan red, tryptan blue, and methylene blue postulates chemoreceptor. So he said that on our cells we have some receptor.
You are saying about morphine.
Sorry?
You are saying about morphine. And continue to next slide. Jump to next slide.
Sorry, your mic has got some problems, so I'm not able to understand what you're saying. Can you write it down? It's better actually.
You are saying about morphine, sir.
Morphine, yes.
And jump to next slide.
I didn't get. What did you say after morphine?
You have jumped to the next slide, sir. Hello.
Yeah, this one.
Yes, sir.
Yes.
You are saying about morphine and jump to the next slide.
Okay. So these, I was just telling about the examples, right? So yes. So I was telling about morphine. So morphine is a painkiller. Yesterday also we discussed it actually. So morphine is a painkiller which is used for treatment of pain, severe pain. Okay. And this morphine is obtained from opium, afim. Opium plant. So you know those flower buds, and then you cut those flower buds, and then the juice comes out of those flower buds, and then you take the juice, and that juice contains morphine. And then cocaine is a synthetic derivative of morphine. So you, by chemical reaction, you derivatize morphine into cocaine. Okay, and then aspirin is a drug which was again isolated from a tree, and then this is anti-inflammatory and analgesic. So which is Disprin tablet you might have used. So this is aspirin, which treats the pain and the inflammation. Is that okay now, Mr. Sethi?
Yes, sir. Yes, sir.
Okay.
Fine. Thank you.
Good. So and then we were talking about, so those were traditional how the drug discovery was done before the modern era. So if you look at the modern drug discovery, so Paul Ehrlich was the father of modern, considered as the father of modern drug discovery. So he won Nobel Prize in 1908. So he gave the theory of differential affinity of biological tissues for various dyes such as tryptan red, tryptan blue, and methylene blue, which postulates the theory of chemoreceptor. Means he said that there are receptors called as chemoreceptors which binds to the drug substance. So this he also gave magic bullet theory, which says that different chemoreceptors for cancer cells or infectious disease, there are different chemoreceptors. And then there is, when you develop a small molecule drug, so it binds to those chemoreceptor, it hit those targets and then treat the disease. So then systematically screened dyes against Trypanosoma equinum infected mice, and he developed SAR and identified trypan red as a lead molecule. So identified trypan red as a molecule which can kill the Trypanosoma equins infection. And then that led to the discovery of Salvarsan, which is the first synthetic chemotherapeutic drugs against syphilis. So in that time, syphilis was one of the deadly disease, infectious disease. And then he discovered this Salvarsan, which was able to treat the syphilis disease and chemotherapeutic. And chemotherapeutic means it was kind of an antibiotic actually. So then there were starting boom in the molecular biology stuff. So there were development in the animal models. So up until 1906. There were no standardized animal models available, and the common house mouse, musculus, was used. So Vista rats model. Have you seen white rats? White rats. So in laboratories, they use white rats actually, and those are called as Vista rats. So Vista rats were used to include spontaneously diabetic rats, spontaneous tumor former, the roster strain, high anxiety behavior rats, low anxiety behavior, the Lobund Vista rat model of prostate cancer, Vista Kyoto rats, an important model for attention deficit disorder, myelin deficient rats, and the spontaneously hypertensive rat, and the most widely studied model of hypertension. So these were some of the animal models which were used for the screening of drugs against different diseases. So nowadays, for almost every drug discovery study, so people use animal models, and those animal models, most common animal model used are these rats actually, rats and mice. So then in 1962, there was another discovery which led to this nude mouse model. So in 1962, so the destruction of FOXN1 gene produced thymus-lacking mice, which does not produce T cells. And so this mouse was unable to reject transplanted tumor tissues and used for cancer research. Because if you are doing cancer research, so generally what happens is whenever you are trying to create a model, a tumor model for cancer research, the normal mouse will reject that cancer tissue because it has its own immunity which can kill those cancer cells. So it was not able to develop a cancer model. But this immunocompromised mice model, so it was, whenever a tumor was injected into this model, so it was leading to increased, it was leading to progression of cancer. So now most of the cancer studies are done in these kind of animal mouse model, where a tumor is grown in in their bodies, and then you try to treat it with certain drugs. And then in 74, this transgenic animal models came in picture. So selective breeding and natural selection of mutations lead to development of transgenic mice models. And then and transgenic means one can insert the gene of humans into the mouse so that the mouse can produce the disease of humans. So this is the idea of genetically engineered or genetically modified mouse model, which can be used for doing research. So many of the diseases such as Alzheimer's disease, where the A beta 42 tau, etc., they are being used for the development of these mouse models and study and discover drugs for these diseases. So, for example, this is one of the mouse model which expresses the GFP, green fluorescent protein, and you can see in dark these mice they glow actually. So they glow. So on left and right hand side there is GFP producing mice, and in the middle there is normal mouse. So you can see that their eyes and their ear, their tail, and their the mouth, everything is glowing, because they can produce this green fluorescent protein so that they can glow in dark. And then there is knockout animal models. Knockout animal models. So where you replace the gene actually. So for example, you in between 6 and 9 you can add new sequence, which is called a sequence replacement method. You replace a part of the gene. So if you want to induce some disease in some animals, so you take the gene of humans and then you add that gene into their animals. And then there is the sequence insertion method, where you do not replace it, but you add the new gene actually. You add the new gene inside the already existing gene. And then there are milestones in molecular sciences, for example, elucidating the mechanistic aspect of drug action or disease progression at a molecular level requires the ability to prepare molecules suitable for testing, and understanding of the structure of the target, and the ability to screen for biological activity in isolated system. So as we discussed, that if we want to develop a drug, we have to test it in the laboratory and then in the patient as well. So whenever we are doing the laboratory evaluation, so the first thing is that you need to synthesize the molecules which are suitable for the target for testing. And then second thing is that you need to understand the structure of the target, like whether it is an enzyme or a receptor, how it binds to the receptor, and how it binds to the other proteins. And then you need to develop an assay so that you can screen the activity of those molecules in a system. So to determine the structure of these targets, so there are multiple ways. So one of them is X-ray crystallography, and this X-ray crystallography was developed by a lot of people. But Dorothy Crowfoot Hodgkin, so she first used the diffraction pattern of a crystalline protein, pepsine, lactoglobulin and insulin in the so for determining the structure actually. So she determined the structure of several compounds like benzyl penicillin, cholesterol iodide, vitamin B12, and insulin. And this is how these 3D structures they look like. And then several developments happened in the molecular modeling and computational chemistry, where, for example, James Hendrickson calculated the conformational energies of cycloheptanes using a computer. So you can simulate the compounds, you can simulate them, how they bind to the receptor in computer actually. And then once you get the a compound, so for example, you have to synthesize multiple molecules. So there are methods, there are ways. Those are called as, for example, combinatorial chemistry or parallel synthesis. This is an example of synthesis of benzodiazepine derivatives on solid support. So you have got the amines and then you cyclize them with these compounds and then you get the benzodiazepine molecules. So in 2013, the number of commercially available screening compounds, it exceeded 21 million. So now this number is even more actually. Around 60 million compounds are available, which can be screened, which can be purchased and screened immediately. So this is a high throughput screening method. I think yesterday also we talked about it. So this is a high throughput screening method where you have all these like computer operated machines, AI operated machines, and then there you start with these plates. These are small plates which contains thousands of wells. So this is a 96 well plate. This is 384 well plate, and this is 1000 something well plate actually. So in each of the well, you add your compound, you add your assay system, and this machine will automatically do everything. So this will add the compound, it will add the protein, it will add the buffer, and then this will go into the reader, and then it will read and it will analyze whether the compounds have shown any sort of activity or not. So in this case, you just need like nanoliter of solution to perform the assays in these machines. And then there is cheminformatics, where you have the use the computers or the AI to design and develop better molecules, actually better lead compounds. Okay, and then we'll talk a little bit about milestones in biotechnology. So this is the picture of Alexander Fleming. So can someone tell me who was Alexander Fleming and how was he famous? Why was he famous? Anyone? Alexander Fleming. I think you should know. Okay, so he discovered penicillin. So what is penicillin? Can someone tell what is penicillin?
Sir, it is the first medicine, first antibiotic that the Alexander Fleming has discovered. I have heard.
Yeah, yes, you're correct actually. So it's an antibiotic. So that is used for the treatment of disease, for infection, treating the infection. So so why he was famous for? Because the penicillin was discovered by serendipity. Serendipity means so what he was doing is like he was culturing the microbes, and then in petri dish plate he was culturing the microbes, and then one day he just kept those plates in open area, did not cover them, and after a couple of days when he came back to the lab and he saw that there were growth of a mold on the plate. Mold is fungi actually, it's kind of a fungi. So there were there were growth of the mold on that plate, and then nearby that mold, there was no growth of bacteria. So he realized that that mold was, producing some sort of chemical which was killing the bacteria. And then he tried to isolate that chemical, purify that chemical, and he found that that chemical was penicillin. So that is how he discovered penicillin, and penicillin was able to, even now it is being used heavily, it was able to treat the diseases like anything, like infections, like anything. Okay, so he discovered penicillin. He also discovered benzodiazepine, which is a central nervous system drug, and also a macrolide antibiotic. So he developed, so there are new animal models. They are based on the selective breeding, and then protein production. That is usually naturally occurring in the cells, but then you have to do that recombinantly to produce them outside the cell, or maybe in recombinant engineered bacteria. And then the cellular assays, which are containing the naturally occurring, cell lines. So then this recombinant DNA technology came in 1953 when the Watson and Crick, so they resolved the structure of DNA, the double helical structure of DNA. I hope everyone knows what is a DNA. So in 1956, the DNA polymerase was identified, which can copy the DNA template strand. And then from 1956 to 85, DNA ligases was identified, which was an enzyme to join the DNA end-to-end, where exonuclease removes nucleotides from DNA chains. Terminal transferases, the enzymes capable of adding nucleotides to the 3' end of DNA. So this is how the recombinant engineering developed. And then in 1884, the tobacco mosaic virus was discovered. And then in 1931, cultivation and isolation of bacteriophages. So these bacteriophages are nothing but these are bacteria eating viruses. So they were discovered, and the merging of DNA modification technology and viral biology to provide a method of artificially transferring genetic materials from one species to another. In 1972, methods of inserting new DNA into simian virus 40 was discovered. So this is, how people nowadays are using proteins in another cell, in bacteria, for example. So what do you think, like whenever you're taking up a protein drug, for example, whether it can be insulin or some other growth factor or some other thing. So those are nowadays, those are producing bacteria. So what you do is you take the gene of human, that protein, insert it into the bacterial plasmid and put it into the bacteria. And then this bacteria will start, producing this protein for you. So that is how we can generate large amount of protein using this method. And most of the biological products are being produced like this, even vaccines as well. This PCR I will maybe skip because it may be a little bit overwhelming for you. And then there are some successes. For example, muromonab CD3, which is a biological product that is for the treatment of transplant rejection. And then you have herceptin, which is a drug for the treatment of breast cancer, which is also called as trastuzumab. And then you have remicade, which is infliximab, treatment of the arthritis. And then if you look at the societal and governmental impact, actually how this whole, affecting the drug discovery and development. So Agostino Salumbrino's observation was that discovery of quinine, it is driven by societal needs to deal with the malaria. So he was trying to find a drug, treatment for malaria, which was a societal problem at that time. So he was, in order to identify that, so he was, looking for how we can do that. And when he saw those Quechua people eating cinchona bark, so he thought, shall we test this drug, this plant for the treatment of malaria? And then there were development of modern cardiovascular drugs. It is driven by its major mortality risk. And then if you look at the antiviral drugs like this COVID-19 virus, so these drugs are developed by the requirement of the society, like the epidemic happened, and then we need to treat that epidemic, so they developed it. And then and then there is this Rogaine, means minoxidil, which is treatment, which is used for the treatment of hair loss. And then there was this, a big story about this sulfanilamide. So yesterday you were talking about these regulatory bodies. So that regulatory body was not, established from the beginning. So it only happened because of some problems with the drug discovery process. Then the government or the bodies, they thought that we need to have strict regulation. And that was because in 1906, the Pure Food and Drug Act of 1906, there were no guidance or requirement on safety of drugs brought to the market. So it was like food only. So you grow something and then you treat the people and then you will get the. But what happened was in 1937. In 1937, the company called Messagil Company, SE Messagil. So they were selling the sulfanilamide, elixir of sulfanilamide, and then this elixir was it killed at least 107 people because of the diethylene glycol, which was present in the formulation. So then the investigation was done, and then it was found that this diethylene glycol was not safe for humans, and it was killing the people. So then they tried to make more strict rules that these should be done like this. And then in 1938, the FDA, Food, Drug and Cosmetic Act came, and then the pharmaceuticals were required to prove the safety of new products through animal safety study prior to marketing approval. And then this is the thalidomide story. So maybe some of you know that. So during 1954 to 56, the clinical safety of the compounds needs to be established, but acute toxicity was not tested actually. Animal study did indicate lack of acute toxicity. So in 1960, peripheral neuropathy happened after long-term use of this compound, this thalidomide. And then in 1961, there were at least 10,000 children which were born with severe birth defects. So they were having, the short limbs. So they were having no, legs or arms. So they were short. And then those people were called as thalidomide babies. So those thalidomide, so this was the drug which was used to treat the morning sickness in pregnant women. So when those women were taking this drug, so it was posing the problem to the fetus actually. So after this, so in 1962, this drug was withdrawn from the market, the thalidomide, and then even more strict regulations that came for the development of drugs actually. So some of the lessons which were learned from thalidomide, so regulatory standards for safety and efficacy testing of new drugs candidate was significantly improved. And then theories on protection of unborn babies by placenta is revised. Earlier it was thought that placenta, which is connecting the fetus with the mother, so that was earlier it was thought that it is acting as a a barrier, and it will help to stop the transmission of drugs or infection to the baby, but it was not true. And also it was also identified that the chirality means a lot actually. The chirality was also affecting a lot. So and then the chiral stability became a new checkpoint for the development of these drugs. Okay, so that was, a little bit about, how those developments happened. And now we will talk about a little bit about the biomolecules, like proteins. How do they their structure work actually? So this is protein is everyone knows that proteins are macromolecules which are a major component of our body. So in our body, if we talk about macromolecules, we have like proteins, we have fats, we have the carbohydrates and lipids and proteins. These are three major polymers or macromolecules, and then nucleic acid we have in the form of DNA. So the protein structure, this protein is made up of kind of a sequence. For example, a word is made up of letters, okay, and you can make a kind of how to say a long paragraph or a long sentence by arranging those words, right? So likewise, in our body, naturally we have 20 amino acids. So those 20 amino acids, they act like alphabets, and then they arrange themselves and they bond with each other to make a long chain of a substance, and that is called as protein. And then this long chain, it arranges itself into secondary structures like alpha helices and beta sheets, and then those alpha helices, beta sheets, they turn them into this tertiary structure, and then that forms a quaternary structure. So it is known that the sequence of the protein means the component, means composition, that leads to structure, and then this structure leads to the function of the proteins. Means sequence defines the structure, and the structure defines the functions. Okay, so it was a big challenge. Can we determine the function directly from sequence? Okay, instead of determining the three-dimensional structure of proteins in the laboratory, can we compute the sequence and the structure in computer by using AI, and then determine its function? So if it is possible, then it will help a lot in solving the problem of drug discovery. So then there are multiple ways to experimentally determine X-ray crystal structures. You have, for example, X-ray generator. You grow the crystals, you shoot them with X-ray, you collect the diffraction data, and then you solve the structure. Okay. Then there is a single particle cryo-electron microscopy. So where you take the image of these particles, and then you arrange those particles, and then from those particles you make them reconstruct a three-dimensional image from those particles. And then there is a structural NMR as well, where you use NMR, nuclear magnetic resonance spectroscopy, to identify the three-dimensional structure of the proteins. Okay, so these were experimental methods, but experiments, they cost a lot. You have to pay the manpower, you have to pay the material cost, you have to deal with the instrument running cost and all those things. So instead, is it possible that we can determine the structure by using computational models or deep learning? So there came this 2024 is a wonderful year for this field. So in this year, David Baker, who designed proteins computationally using machine learning and deep learning, and then Demis Hazelby and John Jumper. So these two guys, they work at the DeepMind. AlphaFold. AlphaFold is a company of Google, actually, Google company, and then this AlphaFold company, they developed these models called AlphaFold 2, and then with that model, one can determine the three-dimensional structure of any protein which exists in this universe. Okay, so that was, so let me show this to you. Is it of interest to you, protein structure prediction?
Yes, sir, very much interesting.
Yeah, so what I will show is now, maybe I can show you the structure of how do we do that actually. So let me share this web page. So this is the AlphaFold protein structure database, which is developed by Google DeepMind and EMBL EVI. So in this, can you name any protein for which you want to know the structure? No, no structure. कोई भी प्रोटीन का नाम बताइए मुझे जो आपके माइंड में आ रहा है. Any protein. Protein we know, right? What is a protein? No. Am I audible or not? Hello.
Yes, sir, audible.
Yeah, so probably you know if you can name any protein, give me a name of any protein actually which is there in your body. Or okay, milk. Which protein milk contains? Milk में कौन सा प्रोटीन होता है? Okay, so let us see casein. Casein is a milk protein. Okay, so this is kappa casein, which is a protein from milk, and you can see the structure. So this is how the casein look like. This is a kind of a very poor structure. So better we go for some other protein, maybe like ACHE, acetylcholinesterase, which is one of the protein which not synthesizes, but it removes the it metabolizes the acetylcholine in your brain. So this is one of the protein which is there in your body actually. And then this protein is responsible for a lot of things in your brain, especially for neurotransmission, like transmitting signals from one part to another part. Okay, so this AlphaFold model, so this is AlphaFold database. So this AlphaFold can determine the structure of this protein from just the sequence, and this is the sequence. You can see this is just the sequence of the protein, how the sequence look like, and these individuals are the amino acids. So these are the amino acids which are connected to one after another and to make this whole large big protein. And these guys, they discovered this AlphaFold. They discovered the technology. They developed the deep learning based methodology to determine the structure of these protein molecules. And then with this server, you can determine structure of any protein, not only of human origin, but for the mouse or rat or rabbit or cow or even bacteria. So this is how they have developed it. And you can see here given AlphaFold, how does it work? Then so this is how it works actually. So this is yeah. So this is based on this deep learning MSA representation. So you have a multiple sequence alignment. You take the proteins, you align them one on the another, and then you use deep learning to make the model, which can then predict the structure of another protein. Okay, so yeah, so that was a basic intro about the history and current progress, and then protein structure and how deep learning can be used for determining the protein structure. And then we will talk about Alzheimer's disease and one of our project where we used machine learning tools for identifying new substances which can be developed into drugs. And then we will do some hands-on kind of stuff as well. We will be using some tools like machine learning based tools to perform some tasks. So before that, I just wanted to have a 10-minute break and then we will start. If you have any questions, then you can please ask. No questions? Okay, then we'll see each other 17:15, 5:15. Okay. Okay, welcome back. So now we will talk about one of the projects which we have been working on, the discovery of molecules, small molecule or drug molecules which can treat the Alzheimer disease. So does anyone know about Alzheimer disease? Probably someone of you can tell a little bit about that. Okay, so Alzheimer disease is a kind of a dementia. It means dementia is a disease of the brain where you start forgetting things actually. So you forget the things which, for example, remembering your relatives, remembering where is your home, remembering like whether you have closed the door or closed the lock and all those things. So you can start forgetting day-to-day activities, and in long term you remember the long term, you forget the long term memories as well, because of the neurodegeneration which happens in the brain. And this happens due to so this is healthy brain on the right hand side. This is your healthy brain, and this is the brain which has been affected with the Alzheimer disease. And this happens due to the presence of amyloid beta plaques which are there in the neurons, extracellular plaques, beta plaques, and then you have intracellular neurofibrillary tangles. So these are made up of tau protein which builds inside the neuron, and then the buildup of these beta plaques and neurofibrillary tangles, they lead to neurodegeneration, killing of the neuron, and further leading to Alzheimer disease. So why we were interested in that is because the relevance is increasing day by day. So in 2019 it was 55 million overall throughout the world, and then in 2030 it was it will be 78 million, and then by 2050 it is going to grow till 139 million. And this is because number of aged, because this disease is of aging disease actually. Yeah, Mr. Sethi, you have any question?
Yes, sir. I have.
Please.
So whether placing some item and forgetting that is also a part of Alzheimer's disease. Just like we are putting our speck on head and forgetting that where is the speck.
No, that is, like forgetting those trivial things does not come under Alzheimer, because you can forget things due to a lot of things. Actually, it can be a stress, it can be anxiety, it can be exertion as well. So that doesn't come under Alzheimer, because for being a disease,
how to identify that whether it is Alzheimer or it is normal unconsciousness?
Yes, that is what I'm telling actually. So to identify whether you're suffering from Alzheimer or not is, first you need to perform you go to memory clinic, and then the clinician will perform some memory tests. For example, there are several tests where you will be shown several objects, and then you will be shown several sentences, and then in the end you will be asked whether you can recall them or not. Okay, and not only that, once you perform poor in those tests, so then your brain scan will be done. For example, when someone get cancer, so they do the PET scanning, PET imaging. So likewise, the brain imaging shall be done, and then it will be confirmed whether there is a buildup of amyloid beta protein or tau protein in the brain. So if these two features are present in the brain, so this amyloid beta and tau protein, then only it will be considered as Alzheimer disease, otherwise not. So don't worry. So it's just, it's like normally we forget things when we are like under stress or we are in a hurry or so. Okay. Yeah, but it will be going to be a major problem because of the aging population. So right now we are considered as the country of young people, right? We consider ourselves a young country where the 30% population, or I think 40% population is between 20 to 40. But by 2050, so today is 2024, so by 2050, these young people will age. Okay, so and the growth rate is going down. So what will happen is by 2050 we will be the country of aged people. So that is going to happen. And aging means those diseases of aging, like Alzheimer's disease. So that is why it is relevant to discover some molecules, some drugs which can be used for the treatment of Alzheimer's disease. And this is the time course of Alzheimer's disease, where you can see that the disease occurs at least 15 to 20 years before the symptoms appears. For example, the problem with the cancer, especially internal organ cancer, is that the patient knows only when the disease has reached to a very high severity. So it has already reached to a very high severity, then only the person knows, and then it is by that time it is already late. So the dementia happens. Dementia means loss of memory and cognition. So this happens already at a very late stage. So before that, the disease already has started at least 15 to 20 years. So can we discover some drugs which can treat the disease at an early stage? For example, can we discover a drug which can start working right here in this in this time, at this time, so that instead of treating the disease at late stage, like at this point, if we can we can identify, diagnose the disease at early stage and start treatment, so we can save the life of people. Okay. So then we realized that there is this protein. May be a little bit overwhelming for you, but there are these proteins which interact with each other, and then this interaction leads to stabilization of the hyperphosphorylated tau protein. So we thought that if we can develop these small molecule inhibitors which can inhibit the degradation, which can inhibit the stabilization of this protein, so it can treat the Alzheimer's disease. So I'll skip this actually. Yeah. So this is drug design strategy. So you start with we discussed yesterday as well. You start with a high throughput screening to identify the lead compounds, and then you synthesize those compounds, and then you perform the biological assay, and then you go back to the synthesis until and unless you are satisfied with the activity of the compounds, so that you can test them into animal models and in vitro and in vivo models. And if you want to see how these molecules they bind to the structure, so you use this structure-based drug design cycle. And while using this structure-based drug design cycle, so you use you determine the structure of the protein, and that I told in like a couple of slides back. So I told you that we use crystallography, X-ray crystallography, or NMR spectrometry, or homology modeling to determine the 3D structure of those proteins. And then you have, you purify the protein, you determine the structure, and then you design the molecules, and then you do the activity screening and all. So in this, what we did was we screened, we crystallized the protein first. So this is these are the crystals. So these beautiful crystals you see. So these are the crystals of protein actually. So we purified the protein, we set up the crystallization, and then you could see nicely grew into small crystals. So by day 32, we had beautiful crystals of the protein, and then those crystals we subjected them to X-ray diffraction, and then we collected the data, and then we designed these. We identified how these two proteins they interact with each other. So you could see here, like this is one protein in the green, and the other one is in yellow actually. So the green and yellow proteins are interacting with each other, and this is how they are interacting. So that we identified using X-ray crystallography. So then we did some further studies where we saw that these amino acid residues, these are shown in like maroon color. So they were, occurring repeatedly, means they were conserved throughout their family, and other amino acids were, they were not that much conserved. So they were like very variable actually. And then we did molecular dynamic simulation on this as well, so we could see that it was interacting nicely with the partner protein. And then by using this structure, so by using the structure which we had determined, so what we did was we screened a library of compounds. So around 300,000 compounds. So we doped them, we screened them against the target, and then we identified 80 molecules, small molecules, which will be further tested and evaluated. So on the other hand, parallelly, we developed this algorithm. We developed this protein purification method, where we purified all those TPR containing proteins which were there in the library. So we had at least 20 human tetrathecopeptide repeat containing proteins in the library, which enabled us to counter screen and estimate the specificity of the molecules which we discovered. So this is a kind of alpha screen assay called alpha screen. It is developed by Perkin and Elmer. So what you do in this assay is that you have a donor bead and then an acceptor bead, and then this donor bead is bound with one protein, acceptor bead is bound with another protein, and when they interact with each other, and if they are close enough, so they send a signal actually. So this donor bead will send a signal to the acceptor bead. And then this acceptor bead will glow, so it will send out light actually, and that will be read, and then it can be quantified. On the other hand, if there is no interaction between donor and acceptor bead, so you do not see any light, there is no luminescence, no light, and then you will see no signal actually. So by using this assay, we screened all those 80 compounds against this partner, so how it inhibited the interaction of these two proteins. And then to our rescue, we found that D10, which was one of the compounds, so it could inhibit the interaction of FKBP51 with FKBP52 with the HSP90 at a very small concentration, very low concentration, which is 100 nanomolar, while the affinity for another protein, which is FK52, was more than 100 micromolar. So it is almost like 1,000 times selectivity for FKBP51 rather than FKBP52. Likewise, we had other compounds as well, like D10, C45, E8, which were also showing activity for the inhibition of this FKBP51 interaction with HSP90. So we identified 30 compounds in low micromolar and nanomolar range, and then most of those compounds they fell in a very few structural scaffolds. Dr. Nisha, do you have any question? Okay, and then what we did was we used this, determine the molecule. we did mutational screening, and then we found that if we mutate this Y, is tryptophan to phenylalanine, so it loses the activity. So that was, the major amino acid which was responsible for selectivity. And then we also did some other assays like GST pull down assay, and then surface plasmon resonance assay.
Hello.
Yes. Do you have any question?
Yes, sir.
Yeah, please.
Whether that previous slide is about the DNA structure, or every protein structure is also a form of helix representation.
This one or this one? Which one? Yes.
Yes. This one.
Okay. So what are you saying?
Whether it is a DNA structure.
So sorry, I couldn't understand. Hello. Can you repeat your question? Or can you type your question, because I am not able to understand what you said because of the problem with your mic, possibly. Dr. Nisha, could you please mute yourself? Yeah, thank you. So then we did MD simulation as well, and then this is the trajectory, how it binds to the the structure, and then stabilize the interaction, and then we did this HSP90 FKBP51 interaction inhibition in cell-based system as well, where we could see that the compounds, they were able to inhibit the interaction at a very low concentration. And then we also tested them in this MTT proliferation and mitochondrial dye system, where it was shown that increased mitochondrial potential upon treatment for 2 hours. And then finally we tested the effect on neurite outgrowth on Sushi cells, where the inhibitors of this interaction can stimulate the neurite outgrowth in differentiated neuroblastoma Sushi cells. So finally, we did test the effect of compounds E8 on tau accumulation in Sushi cells as well, where was shown that the compound was able to inhibit the tau accumulation in the cells. So in conclusion, we could say that we could use the AI or machine learning based or structure-based drug design strategy to make it possible to find low molecular weight compounds inhibiting chaperone-co-chaperone interactions. And the substantial specificity for closely related TPR co-chaperones could be achieved by using this technique. And inhibition of FKBP51 SSP90 protein-protein interaction can be potential therapeutic drug target against tau-pathine AD. So, and we have identified several structurally diverse, potent, and selective inhibitors of the complex, which are on biochemical and biophysical assays gave encouraging results. And these compounds are in further development state. So which platform is used for simulation? Do you mean molecular dynamic simulation or?
Yes, sir.
Okay, we use GROMAX actually. We use GROMAX.
Whether it is open source?
Sorry?
Whether it is open source?
Yeah, the Gromex is open source tool. It is free to use. Yes. Okay. And then, yeah, so that was it. And then what I wanted you to do is, so I want you to do some hands-on. So if you are using a computer, you're connected to this session using a computer. So what you can do is you can so let me show it in the. So can you guys go to this link, tinyurl.com/csjmu-gnina. So those who have the possibility to do it, I would suggest them to perform it themselves. Like what you have to do is you just need a Chrome actually. So you will open this link in Chrome and then follow what I'm saying, follow the instructions, so that you will be able to run some, for example, molecular docking exercise using deep learning. So how we can use deep learning to determine how a molecule binds to a protein structure. So that is what we are going to do. So those who are especially from science background or teaching science students, so they should definitely do it. And those who are from humanities background, they shall also try it, because I believe that this will give you another, a thought direction. So please open this link. So have you opened it? So what you will see is this one actually. Let me share the link. You will see this copy of GNINA tutorial mmv.ipynb. Has anyone opened it?
Yes, sir.
Okay, good. So what you will see is you will see this copy of GNINA, and what you have to do to run it is you have to click on file. Click on file. Let me zoom it a little bit so that you will be able to see it clearly. Click on file and then save a copy in drive. Click on file. Click on save a copy in drive. And then it will show you notebook copy complete. So then you click on open in new tab. So are you able to do it? Open a copy in new tab, and then a new copy will be opened, and then open a new copy in new tab. So then you will see the another copy. So you will have the same, because then it will be saved in your directory, your drive, Google Drive. So how many of you have done it? So because I need a response from you, then only we can go ahead. We can move forward. Has anyone done it? Saved a copy in your drive? No? No one has done it yet? Okay, so what we do in this is like you open a structure of the molecule, which is called as ACHG, 3EY5, 1EVE, 1EVE. So what we will be doing is, so this is the structure of acetylcholine esterase, the enzyme which is involved in Alzheimer's disease. So what we do here is we will use this structure, the three-dimensional structure. Here you can see the 3D structure of this enzyme, and then we will try to perform molecular docking on this enzyme using deep learning. So as you can see here, this is how the Alzheimer drug binds to a protein in your body. This is how it looks like when you look at the molecular level. So this is how the drug binds to this enzyme in your body. That is called as, this enzyme is known as acetylcholine esterase. Acetylcholine esterase. Okay, so what we will be doing is we will be docking this molecule against this target. So simply what you can do, you can just do run time and run all. That is one way to do it. So what it is doing is, it is first installing the required tools. For example, it is downloading the Genina code, which is required for performing the molecular docking. And you can see here it is downloading now. 80% download is done. Okay, and then after installation, what it will do is you will download the file, which is 1EVE, which is the three-dimensional structure of acetylcholine esterase. And then you separate the protein from the drug, which is bound to the protein. And then you use, okay, so then then this now Genina is performing the molecular docking, where it is using the structure of this ligand, and then it is docking it inside the pocket of acetylcholine esterase. You can see the progress is shown by this percentage from 0 to 100, 10, 20, 30, 40, 50, then 100. Okay, so once the docking is done, so you can see here, so this is the affinity actually. This is scoring of different poses, how how different molecules, how different poses, how a single molecule can bind to this protein in different poses actually. So these are the poses like having -10 kcal per mole, -9, -10 kcal per mole affinity, and then this is CNN score, and then this is CNN affinity. So, and this is rank actually, like the pose 1 to 9. So now we will have a look at them by using this pi mole. So in this pi mole, what you will see here is okay. So here you can see that this in the brown color. The brown color one is the one which is the co-crystal structure of the ligand, and then the other one, the green one, is are different poses which are generated by using this docking, Wiener docking. So you can see here now how your drug molecule is binding into the receptor pocket, into the binding pocket, and then this here we have used Genina, which is a deep learning based platform for doing molecular docking and scoring. So this can be used for screening large number of compounds. For example, thousands of compounds can be used to screen against these targets actually. So you can identify potential ligands against these targets. And then in the end, eventually you can download the results like 1EVE, and then you can zip them, and then you can download them. So in your download folder, you will have them as downloaded. So you can further use them. You can visualize them in other softwares as well. So this is how we perform molecular docking using deep learning, called as the tool is named as Genina. So this is one way to perform this drug discovery process. So yeah, that was all. If you have any question, I can take up if you do have any questions.
Sir, hello.
Yeah, please. Yeah.
Whether there is some mechanism present for separating the protein acceptor and receptors. because whenever the accept and recept is complete, then only the disease will be formed. So whether some separator or some form of some algorithm could be specified so that the acceptor receptor can be separated.
Sorry, can you say it again? I don't think your mic has, I think, some issue actually. I'm not able to. Is there anyone who has understood him so they can tell me what he's saying? So you're talking about something receptor and acceptor, no?
Yes, sir. Yes, sir.
Could you please type your question? Is it possible?
Yes, sir. I am typing.
Yeah, please.
Typing.
Yeah, thank you.
Yes, sir. Written.
Yeah, with some mechanism for separating the protein acceptor and receptor in here. Is there in form of some algorithm because all these occurs? Well, yeah. So that is, that concept is the drug target actually. So if you can identify the problems with the proteins. For example, in some diseases, there are certain proteins which are excessively produced. For example, Alzheimer's disease is one of them, where you have this excess of protein called amyloid beta and tau, which are produced in the neurons, and then that causes the neurodegeneration, kill the neurons. So that is one thing, So people are developing kind of algorithms and developing methods which can be used to detect and identify those drug targets, and that we discussed yesterday as well. So yes, there are actually. Okay.
Whether this is a form of sentiment analysis.
Sorry?
Whether this is a form of sentiment analysis.
Sentiment analysis.
Because yes.
It is not like that actually, because here we are more interested into the structure actually. So if there is a small alteration in the structure, then also it can change its function, because as we discussed that this function of a receptor or an enzyme, it depends on the structure. So it is like a lock. If you hammer a lock, so maybe you will not be able to open it with a key, because you have deformed the structure, you have changed the structure of that. Okay, any other question? Mr. Jagat has raised hand. So do you have any question? No? Okay, then so then maybe we can we can wrap up this session. So once again, thank you so much for bearing me for this session. So hopefully, I hope this session has got some some information that is useful for you, and then you can explore this field further, the role of AI in drug discovery study. Okay, thank you so much.
Thank you, sir.